Matter podcast, where we talk about how what you consume, from food to drugs and ideas, influences who you become. I'm your host, Nick Jikomis, and I have a background in academic science, independent research, and writing, and I'm the creator of the Mind to Matter podcast and Substack page. I got a PhD in neuroscience from Harvard University, where I studied the neurobiology of feeding behavior, and before that, I studied evolutionary genetics at the University of Wisconsin-Madison. On normal episodes of the podcast, I talk mainly with scientists, academics, and other experts about topics that interest me as a biologist, ranging from neuroscience to metabolism, evolution, animal behavior, and the effects of drug, food, and biotechnology. This episode is part of a special sub-series where I talk to independent scholars who are often not affiliated with major institutions like universities, but who I believe have interesting perspectives. These conversations are often more multidisciplinary in nature, connecting my areas of scientific interest with subjects spanning social psychology, culture, religion, history, and the rise and fall of civilizations, areas which may sometimes be more taboo, controversial, or outside the Overton window of popular discourse. These episodes are available in full to paid subscribers on the Mind and Matter Substack page or my YouTube channel. If you're hearing this version, you are getting the full episode in its entirety on either Substack or YouTube, and I thank you for your support. Know you already?
Sure. My name is Nick Norwitz. As you mentioned, I grew up in the Boston area, Newton, Massachusetts, little suburb outside Boston. Went to college at Dartmouth, where I studied cell bio and biochemistry before doing my PhD in metabolism at the University of Oxford. And then after I finished my PhD, I came back across the pond, back to Boston to do my medical degree at Harvard. And I'm finishing up the last months of my MD. So about to finish the whole MD PhD saga and go off into the world. And it took me 28 years to figure out what I really wanted to do in life. But I realized I love science, the science of metabolism, teaching about the science of metabolism, and really empowering people to become their own, their own scientists and take control of their health and leverage the power of metabolic health to transform their lives. I've just seen it happen again and again so many times. And if I can have an impact on the world, it will be to make metabolic health mainstream, which is going to, go hand in hand with just empowering people to transform their lives through lifestyle protocols.
Yeah. And, metabolic health has really become a big topic that more and more people are talking about. A year or two ago, I would have said the same thing, but it's even accelerated just in the past 6 months, especially with, all of the social and political stuff that's happening. People have been writing books about this. Politicians have been talking about it, et cetera, et cetera. And everyone realizes, even if they don't have scientific or medical training, I think everyone realizes at this point that something's wrong, sort of systemically with metabolic health. There's problems with the food. There's problems with how we're living our lives, and it has a big impact on our metabolism. That has, that ties into everything from mental health to physical health and all sorts of stuff. My understanding is you also have sort of a really sort of personal story, personal journey in terms of your own health transformations over the years. Do you want to talk a little bit about that?
Sure. So when I was finishing up college and then starting grad school over in the UK, I was struggling with inflammatory bowel disease, specifically ulcerative colitis. For those who don't know what that is, what it sounds like. There are ulcers opening up in your colon. You have bloody diarrhea, in my case, like 20 times a day. It's the kind of condition that, the physical pain's pretty bad, but really it is psychological torture. Your social life disintegrates. You're constantly anxious and stressed. Your mental health goes down the toilet and you just can't engage in life. I mean, in a period of a couple of years, I had gone from a top performing academic and athlete to someone who was withdrawn, scared, my confidence destroyed, and I really could barely engage in academics when I was, there's a point when I was doing my PhD when I was spending a lot of time in the hospital and could not get work done and used to expend so much effort to even go to the bathroom or walk to lab. I remember walking to lab, which was like a half mile from where I lived, and thinking, the amount of effort that goes into this is more effort than I remember putting into my weekly 22-mile runs a couple of years ago. That's the toll it was taking on me. And so it came to the point where I was so desperate that I was willing to try anything. because I had access to the best healthcare in the world. I
Hmm.
mean, I grew up in a medical family, near the Harvard health system and then, Oxford health system and got access to all the best private docs. And
Hmm. What did they say to you initially? What was like the initial diagnosis or how to treat
it was
this?
ulcerative colitis. I mean, like it's not a, uh,
Diagnosis was straightforward.
yeah, you do a colonoscopy, you look
Hmm.
for histological signs, like this is the
Hmm.
diagnosis, this is the
Well,
disease you have. There
yeah.
are some that are like, in medicine, like syndromes and a little bit more hand wavy. It's like, no, you have this disease. Now, it's complicated what causes it. So,
Hmm.
like, risk factors for me included, my age, the fact that when I was a neonate, I had a high fever and I got IV antibiotics. We know
Hmm.
that IV antibiotics when you're a little kid can screw up your microbiome for life and increase risk of inflammatory
Hmm.
bowel disease.
Interesting.
Probably additives to the foods that I've been eating throughout my childhood. I grew up on a, quote, balanced diet, but that included a lot of standard American foods, including Kellogg's cereal, which is now, I guess, a center of controversy. But like,
Yeah.
even
Balanced
if it's
diet
not
meaning like
proven,
the classic food pyramid view of balanced diet.
yeah, well, but because it was a very sensible seeming narrative, especially in my context, because I wasn't a
Hmm.
kid that struggled with obesity. And you think about
Yeah.
obesity as a canary in the coal mine, right? So
Yeah.
it's like, all right, you have this like, young, healthy looking 18-year-old who's like running marathons and has a six pack. And yeah, he's eating cereal and drinking Nutella, but like he's active. So like, he's burning off this excess energy that he's eating. Clearly, like, the cereal and the sugar isn't really hurting him because, he's getting his five a day fruits and veg, etc., and then eating, excess quote empty calories on top of that. But it's not harming him, and we can tell
Yeah.
that because
Yeah. It's the Michael Phelps explanation. It doesn't matter that you're eating, 10,000
right.
calories and it includes syrup and everything else if you're moving that much.
So some people can get away with it for at least a period of time.
Hmm.
In my case, so it's like kind of rolling a roulette wheel. In my case, it caught up with me more quickly.
How old
Some
were you
people
when this started?
21 when I started developing symptoms, 22 when I got diagnosed with ulcerative colitis.
And that's pretty normal for this condition.
That's around the age that you get diagnosed. Yeah.
Okay.
Like young adulthood is pretty typical. Um, so, the thing is though, and this is really important. I don't want people to overlook what I'm about to say. Doctors in Western medicine can only really recommend, quote, evidence-based medicine. And that sounds good. As I just said that, right? Like, of course you want your doctor prescribing evidence-based medicine. And that was the kind of treatments I was going after. But let's unpack what that means a little bit more.
Hmm.
Because evidence-based medicine refers to medicine for which there is evidence. It's not the same as saying we're going to prescribe the most effective interventions because the research ecosystem, the biomedical research ecosystem, is set up to favor pills and procedures. It just is. That is not a judgment on doctors. That is just
Hmm.
acknowledging a reality of the ecosystem where there's a clear business model. If somebody develops a biologic drug that's going to make a pharma company millions of dollars, like they're going to fund those trials. So there's so much more research going into, developing those protocols rather than developing metabolic health protocols. Not because the metabolic health protocols aren't more like as or more effective. It's just the business model isn't there. Again, no judgment on any individuals. We just have to acknowledge the reality of the incentive structure we have.
Hmm.
So I want to use that as a little bit of a framing because, the intervention I ended up using that worked for me, which was a ketogenic diet, a clean ketogenic diet, there are no randomized controlled trials saying that would work on ulcerative colitis,
Yeah.
but it worked in me when nothing else would.
Hmm.
And it's worked in other people. We had a case series on this recently. But point being, I got to a point where I was very desperate, willing to try whatever. And I tried something that wasn't, quote, evidence-based, but had biological plausibility and a strong biological rationale, and it saved my life.
Hmm.
And
And
that drew me into the field of metabolic health.
yeah, I'm guessing you tried other things before this or that your
Mhm.
doctors prescribed certain things for you. What other things did you try that failed to work?
Typical frontline medications, there's things like mesalamine, different biologics, so immunomodulators, those kind of things.
Hmm. Okay, so
And
you
those,
tried.
I mean, to be clear, like those medicines work for some people and
Yep.
they're important and I'm glad they exist and they're valuable tools. I don't want to bash on the innovations of Western medicine. But the fact of the matter is, be it for ulcerative colitis or various mental health disorders, like the treatments we have do not help for a lot of people with chronic
Mhm,
diseases.
mhm.
They're insufficient.
So you tried these evidence-based approaches that do work for some people. They did not work for you. You were desperate. You really wanted to get rid of this issue. As we, it's not difficult to imagine that you'd want to try anything at some point. But there's no trials that were out there pre-existing that said ketogenic diet might work. How did you even think to try that ketogenic diet? How did that come about?
Well, I was just trying a lot of different diets because based on just like logic, you'd think, all right, this is a gastrointestinal disorder. What I'm eating probably should matter.
Mm-hmm.
And I had tried all the other diets that people, would recommend. So specific carbohydrate and low FODMAP diets are like the 1 and 2 for IBD and IBS. And so you try them and they're like, it doesn't really help. At least it didn't help me. Some people it helps a little bit, but I mean, there's so many rules. They don't really like have that big an impact. They definitely don't put your disease into like lasting remission, at least for most people. And then I went through the litany of other diets. Like I did vegan, vegetarian, pescatarian, Whole30, Mediterranean. Like I tried all of these. And then like on the list of things to try was a ketogenic diet.
Mm-hmm.
And that was how I tried it. It was just utter desperation. It's like imagine yourself in my position where you're, depressed, you've lost like, if you're skinny to begin with, and you lost like a ton of your 20% of your body weight in like a matter of months. You're sitting in the hospital bed, like with brain fog, below 100 pounds, emaciated, just like sobbing in the fetal position. And somebody's like, hey, do you want to try a diet for a couple weeks? Let's say there's even a 5% chance that it helps.
Yeah.
You're like, well,
Sure.
sure.
Yeah.
And it turned out that it did. So for me, a ketogenic diet brought my energy back, brought my inflammation markers down to the lowest they'd been in a long, long time, and really restored my quality of life. Now, it was a while before I got my next colonoscopy, but when I did, it showed no evidence of disease.
Hmm. How long did that take? How long had you been on the diet?
The symptoms and the inflammatory markers dropped within a week.
Wow.
It was years before I got my colonoscopy because, like, if you're doing great and you have no symptoms, it's not the kind of thing you just want to book.
Right,
like,
right.
Tube stuck up my butt. But
yeah.
When I did eventually get it, I was in remission. I will say, I want to clarify, this doesn't mean it's a silver bullet and that I don't have any gut issues anymore.
Yeah.
I think for most people with IBDs, like even when you recover most of your quality of life, it's like always going to be like a sensitive area for you, let's just say. So I'm not going to say I have the best gut in the world. I don't.
Mm-hmm.
But I'm here with you talking after finishing my PhD, finishing now med school, like in a happy, fulfilled relationship, like energetic, excited about my career. That is a very different place than sitting in an ICU bed.
Yeah.
So you can draw from that what you want. But that was my personal experience, and it drew me into this area of what I broadly call metabolic health.
Yeah. And so one thing that's important to call out here is you had obviously a very specific need state you were trying to use this diet to help with. You weren't pre-committed to some diet for ideological reasons or because you had a research program that depended on you favoring one model versus another. You cycled through all kinds of diets.
Yeah. Well, it's actually ironic this comes up because it's just coincidental. I was working in a ketone lab. But the PI in the lab in general wasn't pro ketone diet. What happened was back in 2003, the PI of the lab, Kieran Clark, had gotten a $10 million grant from DARPA, U.S. military, to develop. It was something like Project Super Soldier or something. So they were working with my lab with Richard Veach at the NIH, who was like trained under Krebs, Hans Krebs of the Krebs cycle, and they were developing this supplement, a ketone ester that they had done a lot of research on. And I had read some of their work in cell metabolism and thought it was just gorgeous. So, I came to the lab from like a human performance standpoint. And, even in the lab, like my professor, she's like, yeah, and I'm not for the diet. She loved actually like Fig Newton cookies. They were her favorite. I'd always come back because they were named after my hometown. I was on travel to see my family come back with Fig Newton cookies. And like, it wasn't like we were a ketogenic diet lab, but I was in a lab studying ketone metabolism, ironically. But no, I had no ideological attachments. Like even now I tell people if I could have a sweet potato, like I'm not afraid of developing obesity. Like I have a freaking sweet potato.
Yeah.
I would. I don't for therapeutic reasons.
Yeah. And so the ketogenic diet that you started, can you talk a little bit about the specifics there? Was this the classic high fat, sort of adequate protein ketogenic diet? What types of things were you eating?
Yeah. So a ketogenic diet for like the historic one for epilepsy, which has been like very, very high fat and protein restricted, is like a 3-to-1 or a 4-to-1, which means 3 grams of fat for every 1 gram of protein plus carbs. Now, a 3-to-1 diet's 87% fat by fat calories. A 4-to-1 diet's 90% fat by calories. Let me be clear, that is very, very difficult to do.
Yeah.
And also not necessary for the vast majority of conditions. And I was not doing that. So I wasn't protein restricted. I was eating probably 1 gram per pound of body weight, total body weight protein, and probably total like around 80% of my calories from fat. So pretty high fat. On some days I can eat up to 300 grams of fat a day. And my original ketogenic diet actually looked very classically, quote, healthy. Like if I didn't describe it as a ketogenic diet to someone, they'd like, wow, like salmon and like macadamia nuts and avocado and leafy greens, like it looks like a plate of healthy food.
Right.
But it just was very low carbohydrate and put me in a state of ketosis.
Yeah.
So that's what my initial ketogenic diet looked like. Now, I will say I've dabbled in basically every form of ketogenic diet you can imagine, from vegan to full-on lion diet carnivore,
Mm-hmm.
or carnivore. Actually, lion diet's on the schedule of things I want to try. I haven't actually done an official lion diet, so re-evaluating the statement I just said. But typically now I eat a lowish fiber, high monounsaturated fat, high animal protein ketogenic diet. That's
I
just
see.
kind of what I've gravitated towards. But again, not something that I'm dogmatic about in any way, shape, or form. Just what I gravitate towards.
Okay, interesting. And then, one thing that many people would say here, including physicians, including average people, is okay, you're eating this high fat diet, you're eating a lot of animal products, you're not eating a lot of whole grains, you're not eating a lot of fiber, you're not eating a lot of the polyunsaturated fats that have the heart healthy label on the box that a lot of people are familiar with. Isn't this changing some of your blood markers in a way that will indicate cardiovascular disease risk?
Yeah. So, I mean, the first thing I would say is go back to the plate I described for you.
Mm-hmm.
I actually wasn't eating red meat and very little dairy. So if I'm eating salmon, avocado, leafy greens, and olive oil, do you still
That's low.
adhere to the idea that it was?
It's a low saturated fat, high monounsaturated fat diet.
Yeah. I mean, my initial ketogenic diet was around 85% fat as unsaturated, only about 15% fat as saturated. That's basically like a profile like olive oil.
Yeah, yeah, yeah.
You can imagine like my fatty acid profile in my diet was mostly from like nuts and olive oil.
Okay.
I will say like I did have some like, I mean, I had things like full fat dairy and stuff, but like those are also considered healthy and now, don't tend to, quote, adversely affect lipids. So bearing in mind, my initial ketogenic diet wasn't like a carnivore diet, wasn't high in, beef and in butter and tallow and lard and bacon, those kind of foods. What do you think the average physician would expect in terms of my blood markers, good or bad?
Well, for those, I mean, I think a lot of people would consider those to be the healthy fats because you're not
Right.
getting a lot of saturated fat.
Okay. So let's start on that premise.
Yeah, let's start on that
And you can
premise.
ask your next question because you kind of know what happened to me.
Okay, so at this time when you initially started, and that was the particular profile you were consuming, did you get your lipids and your cholesterol and all that measured, and how did that change? If so,
Yeah. So it would be instructive to describe what my LDL, my lipids were before and what I was eating. So before my ketogenic diet, I mean, I ate, quote, the balanced diet, but because of my pre-existing health issues, that I was losing weight and that I was kind of a skinny person at baseline, I was being told to do what I can to eat more calories. So I was literally having my doctors like suggest, including one of my endocrinologists, like, like you want to get a lot of carbs, like sugar's fine, like you live next to a fudge shop, abuse that. So I'd send pictures to my doctors of me like actually eating fudge. You know what? After this, I'll text you pictures in case I can include them in a B-roll. But that's the kind of thing I was eating. So actually quite a lot of junk food, including a lot of saturated fat and trans fats in those junk foods. Now, then my LDL was 90, which is pretty normal.
That's
LDL.
Totally normal, right? Yeah, yeah.
Totally normal LDL. Then I switched to a diet that is generally lowish in saturated fat, proportionally, high in fiber, probably 35 grams of fiber per day, which for an adult male is appropriate or considered appropriate. Main protein source was fish, lots of nuts. And on my first lipid test, I don't remember the exact number, but it was in the low to mid 300s, I believe, for an LDL.
300s
for the LDL alone. Yeah.
And that's so the that would not be what an orthodox expectation would be, right?
Right. Because I was eating I just described what I was eating. Lots of
Yeah.
greens and fish and nuts.
Right. The
So
expectation would be
why
that the LDL should go high if you're eating lots of saturated fat.
Right. So the reason it's important to frame it like that is like, yes, you could say I went on a ketogenic diet, but when you describe what that's like, you have to ask the question, well, yes, my LDL did over triple.
Yeah.
But the question then is, well, why?
Yeah.
It's unlikely the saturated fat was driving it or fiber deficiency or anything like that. So why did my LDL go up? And that was a
Right.
question that niggled at me and is kind of what brought me to studying an area of expertise for me now, which is lipid dynamics on low carb diets.
Yeah. Yeah. And so what maybe one way to approach this is to think about it historically. So let's explain for people like why was this the orthodox explanation that high saturated fat should drive up your LDL and therefore it's mysterious and weird that you had a high LDL because you weren't eating a lot of saturated fat. Where did that idea come from?
Well, it's not an inaccurate idea insofar as if you take the general
Yeah.
population
Yeah.
and you feed people more saturated fat. So let's say I load somebody up on like butter.
Yep.
Their LDL will go up.
Yeah. On average, that's normal. Yeah.
Yeah. And it has to do, with a few things, but like, down regulation of LDL receptors at the liver. So you're not clearing as many particles, LDL particles, and your LDL goes up.
Hmm.
However, one thing to comment on is like, it does not go from 90 to 350 or whatever.
Yeah.
Like
350
the
is
magnitude
really,
of the
really high.
increase is like, it goes up,
Hmm.
but this is a different
Yeah.
class of increase. It's like me jumping versus getting on a rocket ship and shooting to the
Right,
moon.
right. So maybe to give people an anchor point there, what's the approximate LDL level where a doctor would start maybe asking you about taking statins?
It depends on your background profile. So there are
Yeah.
just like algorithms for this. So
Hmm.
like if you have like diabetes or family history of heart disease, they're going to have a
Hmm.
lower threshold.
Yeah.
But let's just say for the sake of simplicity, like in an older adult, maybe with a family history of cardiovascular disease, like say they had diabetes, like I mean the goal if they had diabetes, I think it's below 70.
Hmm.
I'm not great at memorizing the algorithms. That's obviously not my thing, but like 70, 100. Typically now it's like people seem to be going with the lower is better concept, which I'm not personally a fan of, but like, you know, let's just say 300 is the kind of level that would give many doctors a heart attack.
Yeah. It's an astonishing number.
It's an astonishing number. Not even the highest mine has gotten, but it is an astronomically high number.
Hmm.
Yes. So
And
yeah, no, I mean, statin therapy, probably combo therapy would be what people would recommend for an LDL that high. When it gets even higher, as high as mine's gotten, I mean, like for kids with FH, you do like lipoprotein apheresis, like you just
mhm,
like literally rip it out of the blood, like mechanically.
mhm.
Uh,
And there has been for a long time a known relationship between LDL and saturated fat intake, and there is a relationship between
yeah,
LDL, especially when you dig into the details a little bit more, and cardiovascular disease. Can you give
yep,
people just a sense for what is that relationship exactly?
right. So, LDL particles, or the broader class of particles, which we'll call ApoB containing particles. So there's an apolipo. So an LDL is a particle. Like
Hmm.
it's a sphere circulating around in the blood that's composed of several different things that includes cholesterol.
Hmm.
Um, and there's a protein on it called ApoB. And there's a broader family of proteins that also include, ApoB. So LP little a will be another one that maybe people have heard of. It looks like an LDL and has a little tail. But these LDL particles, the ApoB containing particles, are part of the causal pathway for heart disease. So, another way of saying that is they in part do, quote, cause heart disease. And I want to emphasize that because I think it's a point of confusion.
Hmm.
Because there's a difference between saying something is causal and part of a causal pathway versus saying in all cases this is a pathogenic bad thing that needs to be addressed
Hmm.
in all patients.
Hmm.
You know, it's not a perfect analogy, but like an analogy is like, tires are causal for car accidents. Like you need tires to drive the car to get in the car accident.
Yeah.
I mean, you should slash your tires, and I understand reasons why this could be like, an analogy people would pick at, but you get my point.
Yeah.
It's
Yeah.
like I can fully acknowledge, like I'm not denying that these particles have a role in the development of heart disease.
You're just saying it's not the be all and end all for all forms of heart disease for all people.
I'm saying context matters. I'm saying you
Yeah.
can't take a biomarker in isolation
Hmm.
and just decide, this needs to be treated irrespective of context. It
Hmm.
is
Hmm.
context dependent. The risk associated with a given level is context dependent
Yes.
because you have to do an intervention to change a biomarker. Interventions always come with other effects, like real world interventions always come with other effects. It's not an abstraction where I can just say, hey, look at my lipid panel. My LDL is 350. Let me snap my fingers and just change a singular biomarker. It's like, no. For me, the options were pharmacotherapy. We can go through each of them and their potential drawbacks and pros,
Hmm.
or lifestyle change. So in me, you can imagine that as, oh, like adding back carbs. Well, that's going to send me back to the ICU. That's no fun. So it's just, a conversation I want to have that I think we have to have at a high level where we say, look, we can acknowledge that these things can be independent risk factors, but we actually need to get a better sense of what is the absolute risk in any given circumstance and what are the relative pros and benefits of actually addressing this biomarker.
Hmm.
So that gets to another discussion, which we've been circling around, but like the etiology, the causes of high LDL.
Yeah.
Because it can be you have a genetic dysfunction, which I do not appear to. I have totally normal LDL at baseline. This isn't a congenital disorder. This isn't familial hypercholesterolemia where kids are born with levels this high. And kids, when they're born with broken LDL receptors, virtue of a genetic dysfunction, they do get heart disease at a very early age. But again,
Hmm.
if that's a different cause, a different physiological driver. Or you could have somebody with metabolic dysfunction, obesity, diabetes, who then is eating more saturated fat and stuff like that. Their LDL can go up. It won't go as high as a genetic dysfunction or
Hmm.
as my LDL, but it'll go up. And just the point I'm making is it's for a different reason.
Right.
And then you have people like me, which is actually a growing population of something we'll define called lean mass hyperresponders, where I have these crazy, crazy high LDL levels. But for a different reason than any other population known to date.
Hmm. Hmm.
Appears to be a metabolic response to carbohydrate restriction, where my body's fuel sources are shifting, and the high LDL, along with actually high HDL and low triglycerides as part of a pattern, is an epiphenomenon that emerges from a shift in metabolic status.
Hmm.
So just laying that out, I think it's important because the underlying physiology does inform one, treatments, and two, can suggest something about the absolute risk profile. I really don't think I'm as high risk as someone, say, with similar levels who has familial hypercholesterolemia.
Hmm.
I was at that risk. Like I know my ApoB area under the curve, milligram per deciliter year exposure. I should have signs of heart disease, and I don't. It's been checked
Hmm.
on coronary CT angiography for both calcified and soft plaque, and I just don't.
Hmm. Hmm.
So anyway, I'll
So
pause for a second.
yeah.
Yeah, go
Okay.
ahead.
So to summarize, you go through this diet, it improves your GI symptoms. You're eating, high levels of certain fats. You're not eating high levels of saturated fat. You're lean and otherwise metabolically healthy, but you've got these sky high LDL levels. You see higher than normal LDL levels in people with genetic defects, but that wasn't you. You see it in people with obesity and metabolic syndrome who are eating lots of calories and lots of saturated fat. That's not you, and their LDL levels don't go as high as yours were. In
Right.
any case, so you're this different thing. You're otherwise metabolically healthy. You got these sky high LDL levels. It seems to be a response connected to carbohydrate restriction, and you sort of alluded to the fact that you're calling this a lean mass hyperresponder phenotype, and it's happening in some people. Can you give us a sense for like how common this is and what we actually know about why this high LDL phenotype arises in this context?
Yeah. So, just to give credit where it's due, the lean mass hyperresponder term comes from 2017, from a friend of mine, a software engineer, Dave Feldman, who noted the same thing in himself when he
Mhm.
went low carb, and other people in his family went low carb as well. He was the only one who had these increases in LDL, but they again happened as sort of a pattern, with the high LDL, the high HDL, and the low triglycerides. And he's like, well, that's interesting. What's unique about me as compared to my family members that might predict this response? And then looking around at the community around him of other people going low carb, he noticed this, pattern whereby, oh, weird. It's the lean, typically like athletic and healthy people who are seeing these increases in LDL. So he, observationally coined the term lean mass hyperresponder. And it just kind of stuck because then there was this phenomenon of, oh, me too, me too, me too. This is happening to me as well. I'm lean and athletic and I went low carb for X, Y, and Z reason and my LDL went through the roof. Now, fast forward a little bit, we have a lot more data showing that indeed being leaner is a major quote risk factor for having high LDL and low carb diets, such that if actually you do the set of meta-analyses of human randomized controlled trials and you cluster them by, say, BMI category, if somebody has overweight or obesity, LDL does not tend to go up. If they are in the lean healthy range, LDL does tend to go up.
Mm-hmm.
And actually, there's a dose response effect whereby the leaner you are, the higher LDL tends to go.
Mm-hmm.
Nick, I'm just going to take a pause for 1 sec. I'll be right
Yeah,
back.
go for it. Interesting. So yeah, obviously what Nick is talking about is basically that context matters. If you focus solely on one biomarker at a time, you can be really misled because different people can have high LDL in this case for different reasons. Some of them can have it because they have diet-induced obesity. Some of them are otherwise metabolically healthy and lean. And so the LDL is not necessarily an indicator of poor health or anything. And that's part of what we'll probably talk about next with Nick. But that general point is very important. You can't look at one biomarker and interpret it the same way for everyone because all of these contextual factors matter. Do you have a genetic predisposition to having, say, high LDL levels? Do you have diet-induced obesity or metabolic syndrome, something like that? Or do you have a situation like Nick's where he's a naturally lean person and he's eating this carbohydrate restricted diet? Because those are all very, very different biological contexts. And this is something that's, I think, just become more and more apparent in recent history. Historically, right, LDL was thought of as bad cholesterol and HDL is thought of as good cholesterol. You want your LDL to be as low as possible because it's quote unquote bad, and you want your HDL to be as high as possible, or you don't really care how high it goes because it's quote unquote good. And that's just too simplistic. The relative balance of these things matters. The context in terms of your metabolic health and your other biomarkers, all of these things matter in terms of whether, you should interpret a high number as being a good or bad or a neutral thing. So, it's fascinating and complicated stuff, but, the simplistic historical story that LDL equals bad cholesterol and HDL equals good cholesterol, that's just not a particularly useful way to think about it, at least in my opinion, and I think a growing number of people. So we're going to wait for Nick to get back here, and I'll keep talking, I guess, so I don't have to re-record anything. I think Nick's actually getting over an illness right now, so he's on some medication, and he's had to step away. But Nick's YouTube channel is worth checking out. He does a lot of self-experimentation with himself and his diet, and looks at all his biomarkers and stuff. But as he's already mentioned, he's got this interesting lean mass hyperresponder phenotype. All right. You back with us, Nick?
You're just able to ad-lib in my absence.
I was ad-libbing, yeah.
All right.
I was
Hopefully that won't happen again, but sorry.
Regurgitating much of what you said.
Tumultuous day. It's always a good review. So, yeah. So we've characterized this lean mass hyperresponder phenotype. We have several studies documenting the inverse association between LDL and BMI on low carb diet. So the leaner you are, the higher your LDL goes, and that presents, a curiosity. Like, well,
right.
Why does this occur?
It used to be. I mean, it used to be historically, right, that high LDL is basically synonymous with bad stuff, bad health, more obesity, right, etc.
Like, I'm doing something wrong. Like you get that, like, vibe from, doctors or patients where it's like, like I have to improve my lifestyle because my cholesterol is high.
Mm-hmm.
Right? It's almost like a judgment. There is like a moral judgment that comes with having, let's say, obesity, like, being unhealthy in general, and having high cholesterol. Like, people will talk about it in that, form.
Yeah. I mean, LDL has literally been equated with bad cholesterol historically.
Oh, yeah. We had I don't know if I'll get in trouble for saying this, but I'll just say it. My first year of medical school, we had a 7-minute lecture, and it was titled "Why LDL is Bad Cholesterol."
Mm-hmm.
I was not the only one that, when they saw that title, just smashed their head into the desk. But that aside, that is the perception. Mm-hmm. That, like, LDL cholesterol, it's a bad thing. If it's high, there must be something wrong with you.
Mm-hmm.
End of story. So, but that kind of rubbed against what we were seeing, where it's like, well,
you got these otherwise completely healthy people.
Yeah. But it also presented, so we can go into the model behind it. But I'm going to skip over that and return to point out that with different physiologic understandings of the physiologic drivers of these phenomenons, you can test pretty bold hypotheses and generate more provocative findings. So one example people, listening may have probably already heard about is my Oreo cookie experiment, where like based on understanding of why this occurs, and we can explain that in a moment, my prediction, which counters what one might expect, is I could use cookies, in this case Oreo cookies, to lower my cholesterol. Which again, like we place this moral judgment on LDL cholesterol. So like if it's quote a bad thing, then lowering it should be good, which
Hmm.
means you shouldn't be able to use a bad thing to do a good thing in health. So
Right.
why would adding bad cookies do a good thing to your cholesterol? But what I ended up doing in a crossover trial is showing in me, Oreo cookies are actually much better than even statin therapy, so standard of care drug therapy for lowering
Hmm.
cholesterol.
So another way of saying this is you had sky high LDLs on a very carb restricted diet. In other words, you have this lean mass hyperresponder phenotype, and therefore adding something with carbohydrate like Oreos, which would otherwise be
Mhm.
considered just a total junk food, was more potent at reducing your LDL than taking a statin.
Yes, it was twice as potent actually, and was effective in
Mhm.
one third of the time frame.
And in any other context, if you took a statin and your LDL went down, most physicians would say that's definitely a good thing.
Right. And so I'm not commenting, I'm not putting any value judgment on the statin response, which was a 32.5% reduction. But if
Mhm.
the outcome you care about, if you're myopically focused on LDL as a biomarker
Yeah.
or associated ApoB, well, the Oreos were twice as good.
Yeah.
So,
So now you're in this bizarre position of like, okay, how do I rationalize that Oreo cookies were this potent at making a quote unquote good thing happen?
right. Well, from a third party perspective, that's exactly what the tension I want to create. I'm like, it's not really
Mhm.
my job to, well, I'm going to try to explain the mechanism we think underlies it. But like when you have what appears to be a paradox, your job is to express curiosity and want to learn more rather than
Yeah.
place value judgments. Because no, I'm not saying Oreos are a health food. I'm not saying statins are bad. I'm not saying anything about
Yeah.
LDL cholesterol. I'm saying this is a result that I expected. And if you didn't expect this,
You should be scratching your head.
you should be scratching your head and want to learn more.
Yeah.
So the model high level that we have explaining the lean mass hyperresponders is called the Lipid-Energy model. And what it says is when lean, insulin sensitive people who are typically very good at shifting into fat burning mode,
Hmm.
better than people say with obesity,
Hmm.
when they restrict carbohydrates, they have little fat cells, and those little fat cells release fatty acids. Locally, that can fuel some muscle tissue, but you end up with a lot more fatty acids in your bloodstream, like a spillover, you can say. And you need to traffic these around the body again. They need to get back to the fat tissue. They need to get to muscle tissue to fuel muscle tissue all over your body. It's a very like complex choreography with a lot of proteins involved. But again, at a high level, what can happen is the fatty acids get taken up by the liver and then resynthesized into the storage form of fat, which is called triglycerides. Little three fatty acids get put on a glycerol backbone, looks like a little M. But this fat doesn't really mix with water well, like oil doesn't mix with water. So you need a shipping particle, a boat, so to speak. And that's called a VLDL, or very low density lipoprotein. So your liver makes these VLDL particles. It puts the fat in the core to be trafficked around, and around the shell, the surface area, there's cholesterol to help give it shape and form. And it ships it out of the liver, and it goes back to fat tissue and drops off the triglycerides stored fat at fat tissue to replenish the fat tissue, drops off triglycerides stored fat at muscle tissue so muscle can burn it. Then what happens is as the VLDL are dropping off the fat fuel, the triglycerides, it shrinks because you're sucking the core out. So you reduce the volume of this sphere and it becomes a smaller sphere. What does the VLDL become? It becomes an LDL. So your LDL will go up. Now, that's one part of the triad, the high LDL, the high HDL, the low triglyceride triad. The triglycerides and the HDL come in because, well, you're suckling the
Yeah.
triglycerides out from the
So
core.
hold on before we get there. So what you're saying basically is that according to this model, the reason for your high LDL is basically that it's a natural consequence of needing to ship around these fat particles.
Yes,
Yep.
yes. And it occurs in conjunction with other markers like the high HDL and the low
Yeah.
triglycerides
Yeah.
because the
Which would be
protein,
different than an obese person, right? They would
yes,
have a different LDL HDL ratio.
right. They would typically have the LDL would not be as high.
Hmm.
And maybe it could be elevated, but it wouldn't be that high. And the HDL would typically be lower and the triglycerides higher.
Hmm.
High triglycerides and low HDL are part of metabolic syndrome.
Yeah.
I very much do not have. But in lean mass hyperresponders, we see the opposite, super high HDL and low triglycerides, because the protein that turns over the VLDL, it's called lipoprotein lipase, that ends up, generating LDL as a byproduct, transfers, well, it pulls the triglycerides basically out of the core of the VLDL. So triglycerides go down. And then when you're shrinking the VLDL, there's cholesterol on the surface that kind of like falls off the surface and needs to get picked up by something. And it gets picked up by HDL particles. So HDLs will take up some of the cholesterol. So then your HDL cholesterol goes up, and that's how you end up with this triad of high LDL, high HDL, and low triglycerides that we see in lean mass hyperresponders. So that's the model. And
That's
the
the
model
model.
is useful because it makes very particular predictions. So add back carbs, LDL should go down. Hold other things equal, increase exercise and energy demands. LDL should go up. Does it? Indeed, it does. So it makes bold predictions that can be tested. And thus far, it's been pretty on point.
Can you explain that last one? Exercise, more exercise equals higher LDL. Why would that be the prediction?
Because if this is a response to energy demand,
Mm-hmm.
then if you are of this phenotype and you
Right.
just increase the energy demand, you're just going to have the
Yep.
flywheel that is the lipid-energy model spin faster.
Yep. You need more fats to be burned as fuel, so you need to transport them more. You don't have dietary carbohydrates coming in, and therefore you get more higher LDL to transport all of the stuff around.
Yep.
And how common is this phenotype? I mean, when you say lean person, is this any lean person? Anyone with a BMI below a certain level is going to tend to respond this way? Do we
It
know how
would
common
be,
this is?
so we don't know at like the overall population level, it's rare because there is a small proportion of people who are actually eating very low carb diets.
Right.
Of those, very few are lean.
Mm-hmm.
So it kind of,
Yeah.
that specific population is not much. The more interesting question is if you take lean active people
Mm-hmm.
and put them on a very low carbohydrate diet, what percent
Yes.
would convert? And we don't have a value for that. That's
Got
a
it.
study that actually we're in the process of designing. Do it properly. It's a $1.9 million trial, and we're trying to do it properly. So we don't know. I will say there can be many factors involved. I don't think I'd be very surprised if it was 100% conversion rate.
Mm-hmm.
And that's because like these steady state levels don't necessarily inform you about flux through the system. So like some people might be very, very good at increasing their LDL receptor expression on the liver. So they could actually make more LDL particles and turn them over really quickly, but they might be clearing them more quickly than other people.
Mm-hmm.
So, measuring flux is a whole nother question. So there could be different reasons people have different responses. I'm not going to say there's going to be 100% conversion, but right now we don't have an absolute value for that.
Mm-hmm. And so you mentioned this briefly, but I want to return to it. So you're obviously a special case. You had colloitis. You were using this diet to treat that condition. You happened upon this sort of discovery that you got these sky high LDL levels. You unpacked that a bit for us. But is there any concern or any open questions around cardiovascular disease risk here? Would you say like the jury's still out on that to some extent, or that you might be at somewhat higher risk for certain cardiovascular disease events because of the high LDL, or do you think that's very unlikely?
I think that LDL particles are an independent risk factor for cardiovascular disease, part of the causal pathway, but that doesn't inform the absolute risk associated with a given LDL.
Mm-hmm.
And what I would say, or what I would parrot to kind of, say what the average doctor would say is the quote, preponderance of evidence, which is like the majority of evidence, the mountain of evidence we have on this process we call cardiovascular disease, would favor the idea that higher LDL particles is going to drive more disease. Now, the issue with that statement alone is that, well, none of the research projects except one project we did has been conducted in this population. So if you want answers about a particular population, especially a population that is unlike any other population you've ever studied, you need to study that population.
Mm-hmm.
So, in lean mass hyperresponders, there's one study with some preliminary evidence, and it suggests it observes no increased plaque. It was published in JACC Advances several months ago. No increased plaque in lean mass hyperresponders as compared to a matched population with much lower LDL. That does not to say there's no risk associated with the phenotype or the ApoB levels. It's just saying this is a unique population that needs to be studied as a unique population, because it is, And right now we haven't seen any signals. And then what do you do with that? Well, I think at the individual level, like, it's a rock and a hard place situation because you don't want to gamble on your heart health. I'm
Hmm.
not, saying there's no risk. I'm certainly not saying there's no risk. But again, we we have to acknowledge that interventions can come with drawbacks. And so making the choice as an individual can be very complicated because it's not as simple as add back carbs for a lot of people or just take a pill. And a lot of the pills that we offer, we don't know actually either the long-term effects of them. We don't have necessarily long-term safety data, and we definitely don't have long-term safety data in metabolically healthy people. So I'm not going to statin fearmonger, but I'm also not going to say something like, well, we know that PCSK9 inhibitors are going to have no negative effects if you take them for 60 years, because we don't have those data. So you're dealing with, I mean, what is the art of medicine, which is kind of like making a determination with the information available to you while that information is incomplete. And then all we can say is, well, these are the boundaries of our knowledge. We need to be, honest scientists, ask the hard questions and get answers to better refine our choices. So the TLDR version of that is the conservative stance, the conservative perspective is to reduce your risk of cardiovascular disease, even as a lean mass hyperresponder. It seems sensible to lower your LDL and ApoB. However, I won't comment on how much it will reduce your risk, if at all. And that also needs to be taken into the context of like, well, what are you trading for it? Because if you're someone who has to, for whatever reason, trade like, relapse of another chronic disease for some unknown level, maybe minuscule of cardiovascular disease protection, like the patient will tell you, I will tell you it's not worth it. If adding back carbs for me, and I realize it's not a binary choice between, carbs and high LDL, there's other options, but just for the sake of like argument, if adding back carbs for me saved me 5 years of life, but had me having routine IBD flares, I would never ever take that trade.
Hmm.
Screw those 5 years. Like each patient's optimizing for something else, and ketogenic diets, and this is based. I don't think you can argue with this statement. They're potent metabolic therapy for a lot of conditions. Like they are, and so we need to figure out a way to investigate and remove the obstacle to their clinical implementation.
Hmm.
Doesn't mean they're a silver bullet and come without risk, but there are just, you asked about open questions. And one of the things that is an open question is what is the cardiovascular risk profile of people, the minority, who see increases in cholesterol on ketogenic diets? Most
Yeah.
don't, because most on ketogenic diets or trying ketogenic diets have metabolic dysfunction or obesity, and that's why they're trying them in the modern era. But for those like me who do have that response, we don't know the risk profile.
Hmm.
We don't.
One of the things that I think we're going to get into that's sort of implicit in some of what you're talking about is when we think about individual cases versus large scale clinical trial data, for example, a lot of times we look to, what's often called the gold standard double blind randomized controlled trial of some kind in a large population. And, you want your N to be very high in those experiments because you want it to be generalizable. You want it to reflect the population in general. You want to have the statistical power to see certain things. But one of the things that you've sort of said in a way is that a lot of times, when you do those big trials, you're averaging together lots of different types of people, and they can be very different in terms of their individual health history, their metabolic profile, genetic differences, et cetera, et cetera, et cetera. So there's things you can miss about different subpopulations that you don't see by just looking at sort of the average overall aggregate data set.
Yeah.
And so can you start to talk about this a little bit more? Because obviously this is something, you sort of also do a lot of stuff at the other extreme where you're doing this self-experimentation and the so-called N of one experiments. But what are sort of the strengths and weaknesses of large scale trials with lots and lots of people versus taking this more highly individualized approach and looking at individuals?
Right. So, I mean, in a randomized controlled trial, the advantage is if you have groups and you randomize them, either, parallel arms, so you have two separate groups, or crossover, where it's like each person does each treatment, but in a random order, you can kind of account for confounders,
Mm-hmm.
right? Because of the randomization element. And that has its benefits. But again, what you're doing is you're sacrificing specificity because there is heterogeneity in the population. So when you end up with your results and you can say, oh, look, there was a statistically significant difference at a population level, that doesn't mean every patient in the treatment group got helped,
Right.
right? Like you could have a statistically significant difference, but what about all those people who actually can be the majority of people who did not benefit from the treatment?
There could be a subset of people that respond
Are,
very strongly, for
right,
example.
is their biology wrong? No, they didn't benefit. The results are still statistically significant, but
Yeah.
I think, it's misguided to think just because something's an RCT, it's like, a better form of science, or something as a higher N value, it's a better form of science. Those are, as far as I'm considered, like vanity metrics, shallow to some extent. It's all about, is the study you are conducting matched to the hypothesis and ultimately the claims you make? And when it comes to individual care, what matters most is the N equals one. Like an N equals one, like if there's an N equals one study on yourself and an RCT that says something different, the N equals one study trumps that because it's your biology. And again, the RCT overlooks the heterogeneity and individual specificity. So I think there's an advantage to taking this N equals one mindset with the caveats that there are limitations to that approach as well. Sorry, Nick, I'm just going to need to pause.
Yeah, no problem. All right, we're taking a pause here. So basically, what we're getting into are the relative strengths and weaknesses of looking at, say, large scale trials where you've got many, many people, a high N, a high sample size, in other words. And those have certain advantages. You can do randomization to control for certain confounders. You've got statistical power to see certain things that you would not see in a lower sample set. But an inherent limitation of those types of large trials is you're looking at a large group of people, yes, but you often aren't accounting for the fact that different types of people are different. And let's say you have a trial with 5,000 people. Well, if one group responds one way and the control group responds another way, and that's statistically significant, that's great. But that might be because everyone in the experimental condition did respond a certain way, or it could be because 10% of people really strongly responded a certain way to a medication, for example, but everyone else responded weakly or not at all. And so there's lots of things that, there's lots of things that get obscured by looking at large scale data like that. And at the end of the day, I think what where Nick is going with this is that um, you know your own individual biology is, by definition, it's yours. You've got all your little genetic and environmental and life history quirks, and that may or may not correspond to the result you see in a larger sample of people, which is pretty much never going to be a sample of people that are all just like you across all the relevant variables. They're going to be all sorts of different folks. Even if it's all males within your age bracket, say, you're all going to have different genetics, you're all going to have different family histories, and so on and so forth. And so there's just inevitably going to be a lot of cases where, for certain individuals, for certain conditions or certain contexts, the results of large scale trials, may not apply to you as an individual. So we're going to discuss sort of unpack that and discuss some of the interesting N of one self-experimentation things that Nick has done, and the extent, we're going to talk about what do those things tell you that a clinical trial doesn't, and vice versa. What can you conclude with one type of experimentation that you can't conclude with the other? He's done a number of interesting ones that he's promoted on his own channels recently. I think he ate something like 720 eggs in 1 month and did his blood work before and after. He already mentioned the Oreo diet experiment he did, where he actually ate Oreos and brought his LDL down, and we unpacked that a little bit, and a number of other things. All right, Nick, what's up?
Sorry about that. A complicated day. Where were we in the conversation? What was the next?
We're starting to discuss sort of the relative strengths and weaknesses of, large scale RCTs versus smaller scale and even N of one experiments,
Yeah.
such as those that you've done.
Right. So, a couple things on N equals one. Let's just first clarify, like, this is legitimate science. Like, there are top scientists at institutions like Stanford, Harvard, elsewhere, publishing in top journals like Nature Biomedical Engineering, like on their N equals one experiments. One is Michael Snyder. You can look him up, or I can send you his multi-omics papers. Like, it's cool. Like, this can be hardcore, rigorous science, with internal controls, yada, yada, yada. You can get a lot of data. You can do hypothesis testing. You can develop models. Like, certain people have said it's not science. It is. There's no two ways about it. There's nothing that restricts the scientific
Mhm.
process from being applied to an individual.
Can
So
you steel man the counter argument here? What's the perspective of someone says you can't do n equals one science?
I don't think there's a steel man. I don't. I think it's a ridiculous position. Do you want to try to steel man it? And I'll respond to the steel man because I actually think I think it's ego defense. That's what I think it is. I've seen people who are like consider themselves scientists, like esteemed scientists, and I don't know if they're just trying to like it just seems like an ego defense. I don't think it's a defensible position.
I mean, I guess if I was to attempt to steelman this, obviously they're going to make arguments around generalizability and statistical significance. By definition, you can't compare two groups because the N is one, and therefore you can never generalize to the rest of the population. I think those would be like the types of arguments I would expect someone like that to make.
Yeah, but that's not it. So first of all, like you can still do statistics. Like I can collect multiple data points on myself and do statistics and get
Right,
p-values.
right.
Somebody on Twitter recently said on like an article that was published around me and n equals one, like, oh, you can't get p-values. I'm like, yes, you can. Like, I don't know what you're talking about. You absolutely can. You can have controls. You can have pre-post. It's intra individual variability, like what happens before and after. Again, multiple data points. You can do statistics. And then in terms of generalizability, again, is the study matched to the question it's trying to answer? And if the
Mhm.
answer is with respect to individual health, I want there's a trade-off between the specificity and population generalizability.
Yes.
And if you care more about your health,
Right.
the specificity matters. I don't need to generalize. So take it from my perspective for a moment. Like if I engaged in this qualitative n equals one experiment with a ketogenic diet on my ulcerative colitis and healed my ulcerative colitis for
Mhm.
sake of argument, do I care as an individual if that's generalizable, or am I just happy that I'm not dying? Like, and
Yeah.
it's the
Yeah.
same with like many
Yeah.
people I've met with either weight issues or
Yeah.
mental health
And
issues,
and I think
XYZ. Like,
yeah, it's worth emphasizing too. Like there could even, I don't know, it's conceivable, right, that there could be larger scale data out there that might suggest that, oh, well, in this sample of 100 or 1,000 people, they saw no benefit with a ketogenic diet. That doesn't invalidate your N of one experiment for you.
right. So then there's the next level, which is it might not it has might have generalizable elements. So if we do it with enough precision and resolution, what can we learn, especially studying the outliers? So let's say I'm an outlier. I'm an
Yeah.
outlier in many ways. What is it about me that made me respond? And then in understanding that physiology, how can we use it to help more people? And we've done this in history again and again. I mean, talking about lipids, like Brown and Goldstein, they were studying very, very rare genetic conditions in lipids. Does it mean because their N value was only like 5 or 8 that their research was unimportant? No. They were studying outliers with unique physiology from which they learned something. So again, circling back to the main point, is N equals one science? Science? Absolutely. Is there any argument, any legitimate argument for calling it not science? No, not really.
Mhm.
Um,
It used to be more common, I think, in the early 1900s and before, for a lot of scientists to actually do their own self-experimentation, often as sort of the basis to see, whether or not they even wanted to pursue a line of research.
right, right. I mean, it's good for hypothesis generation as well.
Yeah.
And I mean, speaking as the scientist, you can do certain experiments with a lot more rigor, actually more rigor in yourself than in other people. Because if you have a large population of people, say you're like doing like a diet trial or a feeding trial, it's like you can only control what they eat to so much extent.
Yeah.
You're doing it on yourself. Like when I did Oreo versus statin, like I know exactly what I'm eating with absolute certainty.
Yep. You
And I
don't have
can
to rely
actually
on
control
people
it in
filling out a survey or something.
filling out a survey or reporting what they ate or even eating what you feed them
Yep.
or those kind of things.
I think this is
I
an
can
important
control
point
what.
too, because there's also there can be a lot of tricky. So the case of large sample sizes and quote unquote proper statistics can often obscure other methodological deficiencies in the literature. So the entire field of nutrition epidemiology, for example, I'm not going to say the entire field, but much of the field is, large sample sizes and fancy statistics, but it's all built on like flimsy survey data. And so it doesn't actually, those things don't actually matter
Right.
that much.
Yeah, no, but we get caught up in this hierarchy. So you can
Yeah.
come out with, oh, well, there's a meta-analysis
I had a
including
bigger
2
sample
billion
size than you.
people. This happened recently, and I look at it and I'm like, it sounds nice. It goes like, oh, we have 2 million people, and it's a meta-analysis conducted across like 31 cohorts, yada yada yada, the vanity metrics. But then you look at it, and I'm like, But yeah, but look at the data. Two-thirds of your studies show no effect. The overall
Right.
effect size is tiny. There's conflict in the data. The reason for that is diversity in methodology. So you have just not only like you have taken specificity and thrown it into a freaking volcano. Like this does not necessarily represent a bigger truth. It's just, the alternative possibility is it's reinforcing misleading concepts with more perceived muscle.
Yeah,
And when
that's
you
a good way
start
of
to
putting
dig
it.
into like, well, what did actually each study find and why is there variation in the data, you come to very different conclusions.
Hmm.
So I am very much against this perception of a research quote hierarchy because sometimes it's just a way to reinforce pre-existing narratives, by not looking a level deeper at what's actually going on.
Hmm. Hmm. I want to talk a little bit about things that have shifted in the recent past, technological changes, for example, that have enabled this type of self-experimentation where it would have been difficult or impossible or prohibitive to do in the past. So what sorts of tools are enabling people like you to do high specificity research on yourself? I'm thinking
Right.
about things like at-home blood tests and other
Yeah.
devices.
Yeah, no, I'm actually open a kit right here that I just got in the mail. Things like continuous glucose monitor, so you can get real-time biofeedback on yourself. That's
Yep.
now like, approved to be over the counter. You have things like Oura rings and devices where you can get your own data on yourself. I'm partnering with various companies now to like make data more accessible. This was opening this kit. Like this is a kit from SciFox. Ooh, you can't really see it.
Put it a
Eh,
little. Oh yeah, it's a little blurry.
all right. What is it? Well, there we go.
There we go.
Basically like an at-home kit that you just put some blood on a test strip and it's going to give you your ApoB, your LDL, your HDL, triglycerides, HSCRP, your HbA1c, your total cholesterol, homocysteine, insulin, fasting glucose, ALT,
Yeah.
creatinine. Like, you get the idea.
Yeah. I've used
Uh,
I've used that one. It's great. Like you can
yeah, but I'm
see.
saying like there is, a demand for this, and companies are listening and helping to empower people by getting their data in their hands.
Mhm.
There's more and more services to get data, and there's more and more technology being developed. We're going to have, nice continuous ketone monitors soon, probably continuous multi-metabolite monitors. I bet you in 10 years, we're going to have toilets that will analyze your microbiome with every morning movement, and you can do N=1 experiments on your diet and your microbiome health. Like, this is the way the field is trending to get more and more data on individuals and also develop technology to help process that data in the form of machine learning, AI, that'll help us see patterns and understand physiology in an unprecedented way at the
Mhm.
individual resolution level.
Mhm. Um, let's give people one or two fun examples of other experiments you've done. I'll let you decide what you want to discuss in the context of what you think is most useful to maybe help explain some of the concepts we were discussing earlier, things that people will find counterintuitive. Obviously, one that you did recently that I saw was the 720 eggs in a month. But
Yeah.
what have you found there, and what sort of misconceptions does some of that speak to in terms of things like blood lipids, cholesterol, and overall health?
Yeah. I mean, a lot of my recent experiments have had to do with pitting our understand like pitting the lipid-energy model, which like is the model we just went over to conventional ideas around cholesterol. And so one experiment was where I added back a tiny bit of carbs in the context of eating 2 cartons of eggs per day, and my LDL went down. Another experiment I just did, I haven't released it in a video yet, but I did a vegan ketogenic diet, and an animal-based ketogenic diet. So in the vegan diet, I dropped my saturated fat over fourfold. I increased my fiber a lot. I increased my polyunsaturated fat. All those things you'd think, oh, that should lower his LDL.
Hmm.
But I did one other thing that I won't reveal here, but aligns with the lipid-energy model, to see, what would actually happen when I pit this one element of the lipid-energy model against all the conventional ideas. My cholesterol, dietary cholesterol dropped to zero. My saturated fat went down. My polyunsaturated fat went up. My fiber went up. And yet, just spoil the results, my LDL actually went up, not down. Why? Because in this physiologic context, there are more important drivers than any of the conventional things. And so I use these metabolic demonstrations as one, teaching tools about what I consider cool physiology. But I think the broader idea around N=1 medicine is empowering people to get excited about metabolic health and then apply the scientific method to their own life. And that's something that anybody, irrespective of their background training, can do. And, I've seen people, irrespective of language barriers or education limits, apply N equals one science to their own life to great health effect.
Mhm. Do you think what are the potential dangers here, things we should be cautious about as it becomes as it becomes easier for people to do these types of self experiments? How should people think about balancing the advice they might be getting from their physician with their ability to tweak things and experiment on their own?
Yeah. Well, I always think you should have irrespective of your training or background, I still have discussions with my physicians. You should always be open-minded. I mean, that's part of being a scientist, and get outside input, especially from people who have, a greater scope of awareness potentially than you of the field. So that includes your physician. I would never discourage anybody from having a good conversation with their doctor. That said, your responsibility, your health is your responsibility. Like, you should be at the helm of your own ship because you're the one who is most aware of all the variables in your life and in most control of them. So you asked about caveats and limitations. I'd say, nothing that's not obvious. It's like there's the interpretive. There are going to be confounders potentially when you do an N equals one experiment. How rigorously are you doing it? What are you measuring? And did you change other things? So can you really be confident that the intervention you meant to implement is having the effect you wanted? Or maybe there's a placebo effect. So again, there's in interpretive cautions. But these cautions and confounders happen, with in context-dependent factors, really any science. So I think that is a limitation, but I don't think it's a one that actually like truly handicaps the approach. I think not the bigger thing, but the thing that might raise more red flags is the safety element. Like, is somebody going to do something
Mhm.
that is unsafe and actually put them in harm's way? And I think this comes down to just like being a reasonable person. To be very clear, the experiments I do are not those that I'm recommending people do.
Mhm.
I do not go just like eat extra Oreos and expect it to improve your health. That's the metabolic demonstration, not me trying to achieve a positive health outcome for myself.
Mhm.
Um, but the nice thing about like lifestyle interventions is usually pretty benign. Like, if somebody's going to try like a cold plunge or red light therapy or adding more fiber to their diet or even eliminating fiber from their diet or changing this or changing that, like, the risk profile is probably going to be pretty low.
Yeah.
And it will give people, useful data on themselves that I think they have the right to try to interpret and then also seek help interpreting. But the limitations, I don't think, the limitations which we can discuss, and I think we need to have open dialogues about, um, don't prevent this from being an incredibly powerful tool.
Yeah, and I guess even though you're doing more extreme or somewhat provocative things for the purposes of teaching and educating people on this, no, there aren't many people out there who are going to start eating hundreds of eggs per month or do the Oreo experiment. But there have always been people that are people have been trying different diets forever and ever, of all kinds, and it's just really nice now that people within that natural range of self-experimentation that the average person is already doing, trying one diet, trying another diet, there now is the ability to actually monitor your health, like objectively, with biomarkers and with different things, so that you can really see in a way that you couldn't in the past how those dietary and lifestyle shifts are actually affecting your body.
Exactly. And what I would also say is, like, the scientific approach is it's an attitude. Like, people, when they go on diets, the way we conceive of diets in our society now, there's usually like a judgment to it. So like, let's say you go on a diet and you don't lose weight. You might like say, "Oh, I failed this," or like get
Mm-hmm.
angry at yourself, judge the approach, get frustrated, upset. That's not productive. But what if you take a curiosity lens to your lifestyle interventions? Then each of these, you know, interventions are data points, and you don't necessarily get discouraged. It's like, okay, this didn't work. Well, why didn't it work? That's interesting. Let me try something else. And if that worked, oh, why did it work? And is there, underlying thing that, is the driver for why it worked? And can I optimize that? Or can I try something else that like compounds the benefits? being curious about the data that arises from your N equals one experiments generates, a growth and curiosity mindset that is incredibly, incredibly powerful. It's the thing that, like, when I see somebody's eyes light up with that element of curiosity, I know they're going to be successful long term in terms of their health journey.
Mhm.
Again, irrespective of their background, when it's just like, look, I actually find that treating my life as an experiment is a true pleasure, even if it means all the experiments don't have positive results. It's interesting. I'm learning about myself. And over the long arc of time, I am improving. And that's really, really cool.
Mhm.
So I think a lot of it has to do with a mindset shift for individuals. Because like you said, people are doing these things anyway. Like, people are going to be trying diets. But what if you
Yeah.
do it in a little bit more systematic way and with a different frame? Will it
Yeah.
make it a more effective approach?
And if not only do I mean, I agree with you, this type of mindset is very healthy, but in many ways, it's actually a shame that we haven't cultivated this in people earlier. And just to give you one point of reference there for what I'm talking about, I have someone in my immediate family who has had type 2 diabetes, full blown type 2 diabetes for, years and years now, well over a decade. And I just learned, I learned this a couple of years ago, but you're talking a decade, plus 15 years into having diabetes diagnosed, treated, medicated, seeing a doctor every year. I learned that after more than a decade into that diagnosis, this person in my family had never once tried a continuous glucose monitor. A doctor had never once brought it up. And
Hmm.
that's just like the exact opposite mindset of what you're talking about. Is like this is a person who had every reason to use such a device and to get curious, and that was never even part of the protocol, even though there was a particular medical need for it. And like, so if cultivating this curiosity and a little bit more of a self-experimentation mindset was sort of part of the culture already, that would have never even been an issue.
I know. Well, also it's new. I mean, people tend to, like, gravitate, especially old school physicians, to their bread and butter. And when new players enter the field, including devices like CGMs, if they're not familiar with them or utilizing them as tools in their practice, they're not going to bring them up.
Mm-hmm.
We did a pilot study for medical education in my first year, where we used continuous glucose monitors for medical education. And one of the interesting things that we found was that when students had experiences with CGMs, they became better patient advocates and were able to, like, talk to patients with diabetes about the use of these tools, because they were actually familiar. Like, they had tried them on themselves and they understood how they work.
Mm-hmm.
So it made it easier to say, like, hey, this isn't that complicated. This is how it can be a really useful biofeedback tool. Um, and so it removed a barrier to, I would say, physician advocacy. But you can imagine, if somebody has been treating diabetes a very particular way for decades and never used the CGM as part of their practice, when there's an explosion of patients doing self-experiments with their CGMs, it might not be something that a given doctor is, comfortable bringing up. Not to say that they would shut it down if it was brought up, but, that's the ecosystem we're living in.
Mm-hmm.
So I'm not surprised. That happens all the time,
Mm-hmm.
unfortunately.
One thing I want to talk about too is, the public perception. I think the perception here is going to be quite obvious, but the numbers I've seen recently from surveys where they asking people their level of trust in mainstream medicine and institutional science. We've gone from, trust in medical institutions, broadly speaking, being in the 70+% range prior to COVID, prior to 2020. And it's apparently tanked to less than a third now, 30, 31%. So there's this sort of trust in institutions, science-based institutions, that's been in dramatic decline over the past few years.
Mm-hmm.
What do you think is causing that? And what do you think will potentially change it in the next few years?
I think there's been really bad communication around health, and people, the institutions, and using the term broadly, or representatives of those institutions, have done a poor job communicating to the public the rationale for certain recommendations, and certain recommendations have been exposed as having pretty poor rationale. I point to the dietary guidelines, for example, and their evidence base, which I consider incredibly weak. And people are picking up on that because it's the nature of the world we live in now, with access to information, where you're going to have, quote, whistleblowers, or, members of the public who are just going to do a little bit of digging and ask hard questions. And when those questions aren't appropriately addressed, or worse, addressed in a dismissive and defensive manner, people become irate. And you see that now. You see that now a lot. And I think one of the things that exacerbates it, I just said it, but I'll double down on it, is this dismissiveness and elitism. Like some of the comments we saw around N=1 science, like people that consider themselves esteemed scientists saying N=1 isn't even science. I'm like, but it is. Like, if you want to have an open-minded discussion about this, we can. But your dismissiveness is not advancing your perspective. It's just mobilizing people who are ticked off at conventional medicine because historically it's been incredibly paternalistic. And it's been paternalistic in association with people getting sicker and sicker and more frustrated and more frustrated. And you don't need a medical degree to observe that. I'm not going to say conventional advice has caused that, but you can see how the optics are not favorable for a conventional approach. If people are acting pissy and defensive and egotistical and paternalistic, and the population is getting sicker, how do you think people are going to respond? It's
Mm-hmm.
not complicated. It's not like you don't need to be a psychiatrist to figure this out.
Mm-hmm. What is your sort of overall, like, obviously when you tried the ketogenic diet, you had this particular need, this particular problem you were trying to solve for yourself. Ketogenic diets have become more popular among the general population, just as a diet to try for weight loss purposes or to improve metabolic health. Not just in people with something like colitis or anything as severe as what you experienced, but just as a way to improve metabolic health or maybe manage their weight and things like that. What's your sort of overall take on whether or not this is worth trying for the average person who's maybe struggling with their weight, as well as your overall take on how some of the official institutions out there are responding to the rise in popularity of things like ketogenic and carnivore diets?
Right. I would say I think it can be very well formulated and totally safe for almost every individual to try. And if you're someone who's struggling with a metabolic disease or obesity, I think it can be incredibly effective. The literature is showing that, and our physiologic understandings of the drivers of obesity, along with human trials, are showing that there are metabolic advantages to ketogenic diets. So I think it's worth trying. It doesn't mean it is the diet for every person. I'm not going to say that. But if it's something that interests you, I don't think that there are many good reasons to say one should not try it. And again, ketosis is a metabolic state, so you can formulate it any way you want. There are things like, carnivore diets. You could also do a vegan ketogenic diet or vegetarian ketogenic diet if you wanted to. There's many ways to skin this cat. It's just a matter of carbohydrate restricting carbohydrates in a therapeutic manner. So high level, I think that they're becoming more mainstream for a reason. They work, and people are picking up on that. And then my sense is the opinion, or I don't even want to say opinion because an institution isn't a person. The projected positions of institutions as individual entities, and they're not monoliths, but in terms of what they try to or generally promote most in their media, is shifting more and more towards being very open to ketogenic diets. Mm-hmm. There still is a stigma around it for sure, and almost invariably that arises from a misunderstanding and misperceptions and misrepresentations of the diet. Now, bearing in mind, like a lot of the articles that are published and say like have like X University Health News are typically by often by non-experts in the field, not PhDs, not MDs, by like people who like are, cover this thing. And they often misrepresent, miscite. It's often not very good coverage. Or they're coming like from they have predetermined what they want to say because it is comfy and cozy and in line with mainstream narrative. So, a subset of ketogenic diet, or usually ketogenic diet, you mentioned, is a carnivore diet. Carnivore diet is extremely stigmatized. And at this point in time, we're still at the space where people are just going to say negative things about it. If you try to publish something positive about it, there are many, many barriers, I know, because I've gone that far, to getting these things published.
Oh, you're saying there's barriers even in the peer-reviewed literature to getting...
enormous, yes. No,
yeah.
It's not. I mean, the peer-reviewed literature is not a meritocracy.
Yeah.
The standards vary based on like how much your perspective aligns with the mainstream.
Yeah, and that's worth talking about too, because a lot of people think sort of the naive interpretation that one often sees in terms of what the scientific literature is. We want to believe it is a meritocracy. This is the official body of knowledge. The papers published are all good because they were published in peer-reviewed journals, and we can just sort of like take a democratic vote of what the literature says in a given area, and that's the closest thing to truth we can come up with. But there's all sorts of social and political factors behind the scenes that determine the types of questions allowed to be asked, the types of research that gets funded, and whether or not things get published, whether or not the science was conducted rigorously.
Yeah, no, 100%. A lot of it is about who you know, and I mean, it's a peer review process. Your peers are reviewing. So think about the human being who's reading the paper and at a high level saying, do I want this perspective in the literature? And if the feeling in their gut is, I don't really like this angle, let's say, oh, I love a plant-based diet and I don't like carnivore, they're going to find a reason to shut it down. Anyway, no, that's probably a fair representation. Like,
Yeah,
people are
but it's a
giving
generalizable
peer
statement.
review to things that they like and try to shut down things they don't like.
And we know, there's many historical examples of this. One of the best well-known sagas of like this type of suppressive effect is like, back in the days of Ansel Keys, when the diet heart hypothesis was battling against others, there's many examples that we now know of where studies were not published because someone didn't want to rock the boat politically and socially, not because the science wasn't good.
Yeah. No. I mean, and I can come up with pretty extreme examples of like things that get published today things that don't get published that should have gotten published, and there's obstructions to them in various ways, shapes, or forms, or things that get published in quote top journals that
are total trash.
That are utter trash, and you just look at them and your jaw drops. You're like, how did this top journal publish this thing? So speaking of lean mass hyperresponders, I'll give you one example. It was an abstract published in the journal Circulation, which is a top cardio journal, and then presented at an American Heart Association conference. And it was a few paragraph abstract that was something to the effect of, what did it say? Like lean mass hyperresponder stops the statin and goes on a ketogenic diet and has rapid progression of cardiovascular disease. It was like rapid progression of cardiovascular disease. I think it was titled, if you want to Google it, rapid progression of coronary artery disease, CAD, after stopping a statin and starting the ketogenic diet in a phenotypic lean mass hyperresponder. Like, the title was very clear. Lean mass hyperresponder, stop statin, started keto, developed cardiovascular disease. And it's like, okay, first of all, it's a case report. It's an N=1, ironically. So at a high level, you're like, okay, fine. They could probably find one example of this. Like, of all the thousands and thousands, and there are thousands of lean mass hyperresponders, they probably could find one example of this. Okay. Well, first things first. Was it a lean mass hyperresponder in question? No, it just wasn't. They did not meet the criteria for lean mass hyperresponder. In fact, they miscited the criteria for lean mass hyperresponder. But that aside, that wasn't even the appalling thing. When you looked at the case narrative, this was the actual narrative, which was there was a guy who had a prior procedure for blockage of one of the main arteries in his heart, the left anterior descending. He had a percutaneous intervention. Now, this was well before he went on any low-carb diet. So he was on like a standard carbohydrate-rich, mixed macronutrient diet. He had a strong family history of heart disease. He had, blockage of one artery, which they did a procedure for. And while they were doing that procedure, they noticed this other artery is partially blocked. So then they put him on standard of care statin treatment for a couple of years. So bear in mind, during this time, they're not assessing for any plaque progression. They've said, okay, he has disease in one vessel, which we went in and saw. And when we were looking at that vessel, we saw another vessel with disease. So we're going to give him medication and send him off into the world. We're not monitoring him during this time. But at some point, he tried a, quote, ketogenic diet. The diet was not defined. There were no macros. There was no confirmation of ketosis. The patient, there was no duration. The patient literally could have said, yeah, I tried keto, and that could have meant I ate more bacon for a week.
Right. They just took him at his word.
Against the backdrop of being unmonitored in terms of plaque progression on standard therapy, the statin therapy, for multiple years, during which disease could have progressed. And then he had a ST elevation myocardial infarction in the right artery, the right coronary artery, where there was pre-existing disease. So what this narrative actually was guy who was not a lean mass hyperresponder with a strong family history of cardiovascular disease and pre-existing personal history of cardiovascular disease who had built up plaque in this other vessel
Mhm.
that we saw and then treated him with a medication and then went off into the world during which this plaque could have progressed, said he tried a ketogenic diet. We don't know what it was. We don't know how long he was on it. Oh, and then he had another heart event.
Right,
And
right.
somehow this gets published in a top cardio journal with a title that is a bold-faced lie. And that's not even just a one-off. Like things like this happen all the time, and you
Yeah, it's
see
just
it.
yeah, it's just
And until
incredible.
you see the pattern, you just like sound like a conspiracy theorist projecting it. The reason I bring up this as an example is because anybody can Google what I just said, and you don't need a medical degree to like read the title and then read the history and put two and two together and like, wait, this
Yeah, but the unfortunate thing that happens is many people don't go and actually read into what's going on there. Many, including journalists, will simply read the title, see that it's a prestigious journal, and
yeah,
people with credentials are writing this, and then report on it to the general public. And so you can very quickly get total nonsense beamed out to wide audiences
right. No, that happens all the time. There was
it's all done in official
yeah,
journals, official institutions. It seems it should all be credible.
there was a paper that came out in Jack Advances where it was a study suggesting that low carb diets increased major adverse cardiovascular events by like over twofold. And I remember lipidologists jumping at it like, finally, good science. This is like the quote from one lipidologist in Argentina, like, good science on ketogenic diets. And I looked at the paper, I'm like, this is one of the worst papers I ever saw. First of all, the difference in LDL was like 3 milligrams per deciliter, which there's no way, shape, or form that could translate to a twofold increase in major adverse cardiovascular events. Now,
Yeah.
presumably the authors note that because they omitted the absolute difference in the abstract and actually buried it for the main cohort in the supplement, which is kind of like sketchy. And the difference between the populations was also that the quote low carb population had more diabetes, obesity, and smoking. So, like, if you want to
Right.
tell the narrative that, oh, you went
Yeah.
low carb and the saturated fat increased LDL and doubled heart disease risk, like, you can't actually tell that narrative in a plausible manner if you're upfront about what the differences were.
Right.
Because if you
Right.
say, well, we had these two groups and one had LDL, it was 3 milligrams per deciliter, I think it was 3.48 higher. It also had more obesity, diabetes, and smoking, and they had a twofold higher risk of major adverse cardiovascular events. Any idiot can tell you what was doing the legwork in increasing cardiovascular disease risk, even after quote statistical adjustments. Like, and it
Mhm.
was not the LDL, so it was not the saturated fat in the diet here that was the culprit. But the narrative can be crafted in such a way that, well, we're not going to put the 3 milligrams per deciliter in the abstract. We're going to put it somewhere else, and the journal we know is going to be favorable because they're going to want to push forward this narrative. It happens.
So you already have a PhD and you're finishing up med school. What's your plan after you finish med school? How are you going to just apply yourself professionally?
Yeah. I mean, I've recently made the decision to pivot towards some entrepreneurial activities and building up a multimedia education platform around metabolic health. I have to keep a little bit close to the chest, like the exact partnerships and plans I have at this point in time. But I do graduate in May, at which point I will basically be unleashed to do what my mission is that I said it was, which is make metabolic health mainstream. And I think, part of that is empowering people to engage in this N equals one science approach. And then the other part of that is doing it in parallel with advancing the actual science. Because just because I want to empower people through N equals one, scientific methodology, and put their data in their hands does not mean I don't think that like the quote standard rigorous trials aren't important. So I want to do these trials, but the fact of the matter is they can cost millions of dollars. They're hard to conduct, they're hard to fund, they're hard to recruit for. The best way to do that, honestly, for me, is through alternative channels. By having a presence, by approaching philanthropists, then I can actually get the millions of dollars to do the trials I want to do. Whereas I wouldn't be able to easily get them through standard channels, definitely not at
Mm.
my career stage. I would say I'm pretty accomplished for a 28-year-old, but I am a 28-year-old. And I have no interest in waiting 2 decades to be able to pull multi-million-dollar grants and be tied to those when today I can get those trials up and running by being a little bit creative, productively provocative, and I feel privileged to live in, a space and time where I have access to these tools that no generation of scientists or physician before me had.
Hmm.
So I'm just willing to try to take advantage of them in a creative way.
Hmm.
Hence the things that I do. And I won't tell you what exactly spun out of the egg experiment, but it went super viral. It got tens of millions of views across platforms, and it sounds frivolous, but I will tell you, academic collaborations, partnerships, research funding opportunities to the tune of big bucks have transpired out of that.
Hmm.
And if that's what I need to leverage to get the science I want done and empower people to take control of, their health, then I'll do that. And it
Yeah.
might tick some people off along the way, but I'm going to do it.
So basically, what you're describing is, you're by going through non-conventional channels, by doing things sort of outside of the academic institutions that would normally be the channel through which you would raise money to get grants to do clinical trials and the studies you want to do. You're taking a different route, an unconventional route. Do you think sort of this could represent your particular case here could represent something which may be sort of underway already at a larger scale, which is the 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 evolution of new types of institutions that might spin up in parallel and maybe even someday supersede the ones that we have today with all of the limitations that you described.
Yeah, that's an interesting thought. I think one thing I will say is brands are powerful. Like people, like I can tell you, people love to hate on Harvard, for example. Like I see it, and I find it kind of amusing. Um, but the fact of the matter is, they always come back to the brand.
Mm-hmm.
Um, Ivy League, Harvard, Princeton, Stanford, like the brands do have power, whether we like it or not. That could change. But what I've found is they're incredibly resilient. And you'll see this in the pattern of people being like, if something from an institution says something they don't like, they'll like chastise the institution for being elitist, yada, yada, yada. But then if it's like someone like me or someone else at an institution with that brand comes out with something they'll like, they'll happily leverage the brand. So like the brand just gets more voice play and more attention. And I honestly think it only builds the brand. I don't think these brands have been around for hundreds of years. I don't think they're going anywhere. They're not doing themselves any favors, let's be clear, by some of the ideas that they propagate at an institutional level. But I don't think they're going to die within the next like 10, 20 years. Now, the concept of alternative institutions is interesting. I think it'll be hard for them to get traction among the general public. Um, but we'll have to see. I really don't know. It would be nice, quite honestly. I like the idea of democratization of science, more accessible science, especially given the price tags and inaccessibility, irrespective of intelligence and qualifications for getting admitted of some of these institutions. Um, so I don't know that I have a great prediction, but what I will say, what came to mind when you asked the question is I found, for better or worse, the brands are incredibly resilient.
Mm-hmm. Is there anything that you want to reiterate that we've talked about, or any final thoughts you want to leave people with about anything we discussed or anything that you're doing?
I would guess, with respect to observing me, um, and this might have, generalizable wisdom in it, but think about like what to you, this is a term I think about a lot, productive provocation. Because we're living in a time when like, engagement is a currency. And it's something that we just have to accept as a practicality. And, it's a space that we have to compete in. With that said, I think it's up to every individual where they draw the lines on how they provoke, how they use clickbait or legit bait in order to start a conversation they want to have. It's kind of a means to the end argument. And I really don't think it's cut and dry. Um, so I just think it's always interesting to assess what you think an individual's motivation or incentive structure is for doing what they do. Um, because sometimes it can be a lot deeper than you think. A lot of times it's not, but was something like the egg experiment. I'm not going to delineate like every single benefit that came from it, but like I will tell you from a academic and research perspective, it had a ton of advantages that the vast, vast, vast majority of people will not be aware of from reading the headlines, which just
Mhm.
look like a goofy headline. The people that know me and know how I think strategically will see that. And it's something I like to try to communicate with openness and transparency, and honestly, something I'm almost not gun shy, not ashamed, but something of that valence about because I realize my purpose, almost with intentionality, isn't always clear on the surface. Because what I'm trying to do is generate an intellectual ecosystem and capture more people. The reason I'm ending on this thought is actually because I think people can see me struggling with how to communicate it. Because these aren't easy things. But academics, be it me or other people, need to learn how to compete in the media ecosystem because there are going to be voices who are going to be loud and uprising around health, metabolic health, science, etc., policy. And if you want to have a seat at the table, you need to learn how to play the game to some extent. Um, so the game has many players, there are many approaches, but I think it's just worth every consumer being aware that that is the reality and keeping that in the back of the mind where they're evaluating what people are saying, and then deciding, who they want to trust, who they don't want to trust. It's not an easy thing. That's why I bring it up. There's not like a clear black and white. Um, so it's something I'm struggling with as a communicator early on in my development, just because it is such a delicate balancing act. And it's very hard to know prospectively if you're doing the right thing, be it, the egg thing or the Oreo thing. It's like before I do that, I'm like, is this gonna like what is the net effect of this gonna be? There are definitely gonna be negatives. They're definitely gonna be positives. And what's gonna be the net balance? And you can't know until you do it, and then you do like a retrospective audit. Um, so I think about that a lot. I think about the communication styles I use, how it reflects back on me in the short term and the long term, how it reflects on my colleagues. And I will just say it's probably where I put the most neuroticism and anxiety, and rumination because it is such a difficult issue. Like the metabolism is the easy part. Figuring out how to communicate it in a way that can scale, capture attention, but also provokes productive discussion and is nuanced and it can advance people's wellness is it's fun, but it's tricky.
Mhm. All right. Well, Nick Norwitz, thank you for your time, and I look forward to seeing what you come up with next.
I thank you so much, Nick. Appreciate it.